Welcome, dear.
Maybe. You'll just bugle up.
Hey, Bob. Guess who I got a call from yesterday or the day before? Bob. Hello. Hey, Bob. You there?
Bob, I thought I saw you leaving the Arboretum yesterday. But he's muted, so he can't respond. Maybe he doesn't even hear
He's
me.
not answering anyone.
He looks perplexed.
Yes.
Like the speakers aren't working. Bob, wave if you can hear us.
No,
It's not happening.
nothing.
Bob, unmute yourself.
Well, I think it's not working on the other end. Like,
We might have to
now he's
message
typing.
him.
Now he's typing. No. Anywho. Oh, he's
Anywho.
trying to get audio to work.
Oh,
I hope it works.
but he can't.
Brian, as an aside, I heard from the Fairy Woods woman. It was the woman who had been trying to call me.
Okay.
Thank you.
Do you think Bob needs, I could chat with Bob about how to check his audio.
You should do that.
Okay. I'll mute
You need
myself.
one of those grease boards where you can say, hey, Bob, and hold it up to the thing. And
I'll mute myself. I'll give him a call.
He answered my chat, so I'm assuming he's got that down. Hey, Donna.
Hi.
Enhance further immunity from civil liability on the basis of.
hey, Donna,
Howdy, Andrew. How are you?
I'm good.
Hey, Jeannie. All the folks.
Is causing nations in surrounding states.
Peter, you gonna need screen shared or anything?
Here Now is a production of NPR and WBUR. I'm Celeste Headley.
Peter. You.
I tried to call Bob on his cell phone and he didn't answer. He must be busy with something.
I don't know. He jumped up like he was answering it.
Well,
Did you chat him? Because he chatted back. He can do that.
I guess if he puts on the closed captioning, he can see it. We'll
Did
get
you
there.
ask for the live transcription? Brian?
Did he? I don't know if he asked for it, but it's. I just
No,
saw that it came
someone
on.
did.
Yeah, actually, I was in a conference. I mean, I was helping to manage a conference a few weeks ago, and one day the setting wasn't turned on, and we had a lot of people that missed it and really like having that. You know, the subtitles, the closed captioning is helpful.
Subtitles get pretty funny sometimes and they mistranslate what you're saying.
Yeah, I think for plant people, especially when you start doing plant names, it's not ready for that. It's not ready for, yeah, Logostromia. No, that doesn't come up.
Yeah, but it's usually the first couple words of a sentence that the hard of hearing or soon to be hard of hearing miss. So it's really appreciated by those folks. They feel less left out. Anyway, it's what I found.
Huh?
It is a joke otherwise.
Be excited if they could do live, like closed captioning, but also translate it. Because imagine like the translation, if you could like do live translation to Spanish or whatever. I mean, you just like our accessibility would open up huge, I feel like, but maybe that technology is not quite here yet.
Well,
You need
actually,
a supercomputer for that.
it used to be that you had to translate it yourself, type it in yourself. And so that option still exists. I mean, you can hire someone, or you yourself can have a member of the meeting typing it in, translated.
I guess I'm dreaming of the computer just doing it automatically.
Automatically.
Oh, like Google Translate or something.
Yeah.
Sure. Why not dream? That's the way.
I mean, we can dream big, right? Cars drive themselves.
That's the way
Normal
to get
citizens are
ahead.
going into space. We should have translation.
Bob is unmuted. Finally.
Well, it's how many people are in the room? You have 24. We could probably get started. I know, and I'm not sure if I'm a co-host yet. So make me a co-host, and I can get started. And as folks filter in, I won't talk about anything super important for the first 2 minutes. but and then I'll get into the core of it. But luckily, Peter's on here as well. So Peter Fixer's our chief arborist, so he's on here. And you'll see after I talk and Peter talk about our summer swings, Tim and Cindy are on. So they're going to be following up behind me. I think Pam is coming right out of the meeting, so she's not going to do the introduction today. So I'm going to do the intro. So I'm Brian Thompson-Nowak. I'm the Director of Education. I worked in continuing education for a long time. I was an intern, so 10, 11, 12 years ago. So if you haven't met me before, that's who I am. So I'm going to get started on the summer swings. I'm going to share my screen, and people are going to be, and there's Pam. All right, so I'm going to share. All right, so summer of swings, and I'll say this is something new and fun we have planned for the summer. And with the title, you can tell what summer of swings is. We didn't hide it or get too cutesy with it. It's actually a summer of swings. So it's going to be just during the summer months. It's not permanent. It's not in the spring. It's over the summer, and we're going to have swings from trees in the garden. So just a little bit of background on this. When we're thinking about, how do we, for a long time up until the pandemic, we would do something different every year to attract new folks into the garden, to get people to come back. So Patrick Doherty has gone in, and that's been in for what's 3 years now, I think, for Patrick Doherty last exhibit. We've had Adirondack chairs, we've had bird houses, we've had big bugs. I mean, I can go back to all of them. We've had lots and lots of things here to get people to come in the spring, the summer. And when we were thinking about this year, for the past couple of years, spring kind of does itself. We don't really need to get more people here in the spring. I think if any of you can remember back to our Japanese Cherry Blossom Festival from a few years ago, I think we broke Chestnut Hill. We didn't like it. It was too much. So we actually don't need to do too much more in the spring months. We're going to let that be and just let the garden speak for itself, because people will come to the spring. So we're looking at summer. And so what we're looking at for the goals of this summer was, we want people to get excited about visiting the summer months. The summer is traditionally a time when our visitation dips a little bit. Once it gets into the 90 degrees, we don't get as many folks coming. So give folks a reason to come this summer. Give people a reason to stop and look up in the canopy of some really cool trees. There's a lot of great trees here. So give people a reason to stop and look up. We want to educate people always about the trees, about the wood, about anything we can, physics, while people are out there interacting with the collection. One of the things we want to do this year is spread people out, not just have like we've done with the scarecrows. The scarecrows used to be just in Oak LA. Now we're spreading them out to the Arboretum. So this theme of like spreading people out in the garden, spreading things out. We wanted to get folks around the garden. Again, highlight amazing trees. Let people know they can have fun. People can have fun here. It's not all serious. It's not all science, which Tim and Cindy will get to in a little bit. There's still lots of science happening, but people can have fun, and also give people new places to explore. So I've heard time and time again when folks say, oh, I never get to the wetland. Somehow I didn't know I could go there, or I didn't know I could go down to the Magnolia Slope. I didn't think I was allowed to go there. So people have this perception of there's places they're not allowed to go, but really you can kind of poke around the Magnolia Slope. You can see the dogwoods, the wetlands. You can go all over. So we kind of want let people explore the garden a little bit more. All right, get over. All right, so what is it going to look like? So this is a great picture. And for a long time ago, I worked at Bartram's Garden, and just after I left Bartram's, they put up a tree swing. And you would think it wouldn't be that big of a deal, but this swing has been up now for, someone can correct me if I'm wrong, for over 5 years easily, and it's been up for a while. People love it, and it's a great. If you ever get to Bartram's, go and swing in their tree swing. It's down on a London plane tree right down from the visitor center. But we're going to have 10 locations in the garden. So, and I'll show you a map in just a minute. So these are locations all throughout the Arboretum, and all these swings are mostly going to be made in-house. So Andrew and the arborist team, they have wood that they've collected over the years from special trees, from interesting trees, and Andrew was kind enough to use his chainsaw mill and create some benches. for these swings. So we made them almost all in-house. Bill actually took a bunch of these planks up to Maine and put them through his planer and routed the edges. So they were made in-house by our team here. The only swing that's not made in-house is we're going to get like a porch swing or kind of A-frame type swing that you just have on a porch, handmade by a gentleman that does our green woodworking classes. His name is Carl Newman. He's going to make it by hand with his like hand tools and have that over by the swan pond. So that'll be pretty cool. We're also going to do interpretation. We're not just going to put these out there and not have anything around them. We want to have interpretation, interesting things about the trees that they're on, interesting things about the trees that the benches are made out of, the history of swings. It's, you know, doing some research on like the thousands of years that people have used swings to cool off on monsoon season or pick apples up high in the trees. It's some pretty cool stuff. And health benefits of swinging, you know, doing some research about this. You don't realize that there's a lot of coordination that your body has to do to actually swing in a swing. And I think I totally forgot, like the first time you swing as a kid, you have no idea what to do. And eventually you get that whole rocking motion, and it's good for your core strength and coordination. Yeah, I talked about everything else, but then the Galileo in the garden, and I'm not sure if Jeff Clark is on here. If Jeff is on here, so some of you know Jeff. He was a science teacher, if I'm not mistaken, for many years, and he was really excited to share with us his pendulum experiments. And if anyone of you don't know Galileo's pendulum experiments, we're going to be doing the Galileo in the garden. So Galileo did an experiment with pendulums. That is, it doesn't matter how heavy you are on a swing or how far back you pull yourself on a swing, it'll always be the same amount of time to swing back and forth, despite changing the weight of the person or how far back you go. The period will always be the same. So we're going to invite people to test out that theory, change up the size from adult to a child. And as you go from swing to swing, you'll see that the amount of time, the period it takes to go back and forth will be unique per swing. So that'll be a fun like physics experiment that people can try in the garden. I'm going to go next. So I already know that this is going to be fun because Andrew tested one out and we couldn't get the grin off of his face. So there's Andrew testing out his swing. So these are the 10 locations of where the swings are going to be in the garden. And if I say like the core of the garden, we did a study a few years ago where most people go in the garden. And most people go from the parking lot. If you can see my mouse, they go from the parking lot to the visitor center to out in the limb, past the rose garden.
Brian, we can't see your screen.
Oh,
Are we supposed to?
you can't see my screen?
I can see it.
Yeah, I see it.
I
must
see
be
it.
me. It must be me, sorry.
Oh, sorry. So as you can,
Brian.
so most people do this loop that goes past, once you get past the trains, around the azalea meadows, swan ponds, log cabin, fernery, and then they leave. So most people keep to this very inner core of the garden. So what we're hoping to do is get people a little bit further out into English Park, get people around here to the natural areas, the wetlands, and also, which I know is a little bit crazy, on the other side of the driveway behind the parking lot, which I know is people don't go over there too much, but if you know the bur oak I'm talking about, it's an amazing, amazing tree.
Hey,
So
Brian.
that's where we do yoga
Right.
in the afternoons for yoga classes. And it's also a good wedding site, so you probably know that. But there's also a nice giant dogwood over there as well. So that's the 10 locations.
Brian.
The chinkapin, the Henry chinkapin, and the Oriental plane tree. And if you can, so you can see this. This is a kid swing. So these smaller trees, the chinkapin is going to have a kid swing. So it's not going to be a full. If you know the chinkapin, it's not a huge tree, but it's a decent sized tree. So that's not going to have full size adults on it. But all the rest, besides the Oriental plane tree and chinkapin, will have kind of the standard bench swing that you saw in the picture before for here. And Peter, I'm not sure if you have your hand raised to jump in.
I do. I just
Yeah, jump
want
in.
for the volunteers to let them know if they do wander off looking for the bur oak. This map isn't a great representation of where it is. Really within a stone's throw of the giant dogwood and closer to the Meadowbrook fence. So don't take that as literally where it is. It's definitely closer to the giant dogwood.
Yeah, well, if you start to go that direction, if you look for a large oak tree, you'll see it. So yeah, these are I think Liza did this, but this is a general reference of where it is. And one of these cool trees, this Chinese chestnut, is right on. It feels like you're right on the edge of the driveway, but that's going to be one that no one really walks down that kind of you're just like just outside the trail from when you're going down from Widener down to the wetlands. If you were to pop out the side, there's a couple of Chinese chestnuts there that are along the side of the driveway. So these are, and I'll let Peter speak to all these trees and the vetting of the trees, but we had a lot of trees that we had initially thought of as good options, and these are the ones that Vince and Peter, Bill thought would be the ones that could actually handle, be able to handle a swing on there with adults. So let me look. I think I was going to pause here and let you guys ask some questions before I get into Peter, the kind of explaining how he's going to hang these. He has a whole really unique system of hanging them, making sure they don't damage the trees. And just so you all do know, we're going to put mulch down to protect the area underneath the trees. We know they're going to get some wear, some visitation to these. So we want to make sure we can do as much as we can to keep mulch down for safety and for the root zone of these trees. So I'll pause and let you guys raise a hand or just shout out.
I have a question.
Sure.
So will the visitors have to sign some sort of Mars Arboretum, save Mars Arboretum harmless from injury?
No, I mean, right now, each swing will have a sign that says, parents and guardians, please watch over your children. This is, it's a playground or playground type equipment, use at your own risk. So there's not a waiver, similar to if you were to climb the Sugarman sculpture, and fall off. So any misuse of these is at your own risk.
Okay.
All right, I'll go to the next slide and let Peter kind of describe a little bit of his tree protection methods that he's going to be implementing.
Okay. So what you see in front of you in the left slide is what we're going to have the rope laying across. Instead of having the rope directly onto the lateral branches of the tree, the load's going to be spread across, depending on the diameter of the branch, a number of, I guess they could be 1x2s with a little notch in them to hold the rope in so it doesn't go off in either direction. And that first is going to be, you can see by the black line, kind of tied to the branch, and then the rope's going to be wrapped around it, and we'll be able to, with using the U-clamps you could see in the picture on the right, we'll be able to fine adjust the height of the swing left or right, up or down, so it's nice and level for the swing to be established on. And we'll dog those U-clamps down real tight with some Loctite on them, so there's no risk of them coming undone during the summer months. And if the question of concern might be, what about secondary growth and girdling the tree? These are only going to be up for 3 months, so that threat is not very real, and the load being spread over the 1x2s should alleviate any wear. The place I got this idea from is having installed zip lines at places. You usually use kind of narrow diameter wire, which they wrap around the stems of trees for their anchor points, and you don't want that narrow gauge wire digging into the stem of the tree, so they would always use little 2x4s to spread the load out around the stem, in which they put a lot of force on to hold the wire taut from tree to tree. So that's the origin of this idea, and we've tested it out a little bit. It works very good. We haven't fully installed one, but so far so good, and that's where we are.
Peter, can you speak a little bit just to like the general trees, like the size of the trees that be able to handle this amount of weight?
Oh yes, Brian said, we vetted a lot of trees, and trees that best suit the bill that were very mature, very large trees with large diameter laterals that could handle a load of well more than we could put on with 1 or 2 people hanging on the branch. So that was, limiting factor on what we were seeking and what we decided upon using. So I said to Brian, these are trees, these are tree limbs you could hang a truck off of. So we're not really any fear of them failing with whomever may want to swing on them.
And I'll also say Peter and Andrew right now are going through all the trees doing the crown cleaning. So some of these trees they're tucked back in a little bit. So yeah, I know I can let Peter speak that out.
Yeah, thank you. There, our kind of goal right now is to go through each of the trees proposed to have swings in and their adjoining trees to prune out any hazardous deadwood that could potentially come loose to a higher frequency of visitors being underneath them. So we don't want to have any mishaps happen just from a dead limb falling out of a tree because of a higher frequency of visitation in that area. A lot of these trees, they're great to look at from a distance, but they might not invite people to just hang out under them, let alone shake the tree around by swinging on it. So I personally am going through each tree and what I will call pencil deadwooding. There's not so much as a pencil left of deadwood in the tree when I'm done, so that nothing really has a good chance of falling, even being an annoyance to somebody, let alone a potential injury.
And I think we're going to do, I think our minimum inspection by the university is monthly, but we'll be doing it more often than that. So the whole time these are up, we'll be doing inspections to make sure nothing comes up. The ropes are not wearing, or the branches, there's nothing happening to these. So we'll be inspecting them throughout the summer. And once the summer's over, the swings will come down and we'll jump right into the fall season. So it's a temporary exhibit, and we're excited to get ready. I mean, right now the swings are being laser engraved with the type of wood that they are. We have angler beech in there, which is cool, and some eastern red cedar. It was interesting to see what Andrew and Peter had in the stockpile of wood. So there's some really interesting, cool wood and grains that are going to be out there. So it's unfortunate that some of these trees came down like the angler beech, but it's cool we can see them in bench form, and so they'll be laser engraved with the type of wood that they are, and you'll be able to see the different graining. So that's something else you'll see out there.
We're really gonna seek the best formula in terms of, for example, like linseed oil or whatever we may use to really highlight the wood color and wood grain of each seat.
Yes, I'll stop screen sharing. If folks want to ask any more questions, while Peter and I are here, Liza's here too. Liza and I, and Jan McFarland before she retired, and Jeff, we've been working on interpretation for this for, I guess the past, it feels like the past year and a half, but it's probably been about 6 months. So we've been working on interpretation for this. So that's going to be, we're getting that set up and ready to go. So this will go out, like I said, the first day of summer is the June 21st. So right as kids are kind of getting out of school and the temperatures are starting to go up, then we'll put this out.
It's gonna be a lot of fun.
Yes, if there's, and of course, I'm available with email if you have questions between now and then. But if there's no questions now, we can pass it off to Tim and Cindy.
Thank you.
And I will pass it off to Cindy.
Hi, everybody. Can everybody hear me? Hi, Pam. All right. Let me see if I can share my screen here. I am disabled to share screens. So does somebody want to make me a co-host?
I did, you're on.
Okay, great. All right, can you see my screen?
Yes. Yeah.
Yes, thank you.
Once again, I cannot see the screen that's shared. Did I perhaps not click on an OK to record something, or? But I couldn't see Brian's maps and I can't see Cindy's screens. Sorry. But just keep going. Although I think we need to mute a few people that keep popping up as speakers. Jeannie and Al Schreiber and Al Gale, Andrew Gale.
All right, I'm going to go ahead and start. If somebody wants to mute people, I'd appreciate that. So for those of you who don't know me, I'm Cindy Schema, and I'm a botanist here at Morris. And thanks to Pam and Liza for the opportunity to speak. I'm always glad to talk to the volunteers here at Morris and share a little bit about what we're working on in the botany department. So today I'm going to talk about a plant that's called Virginia mallow. That's the common name. And the scientific name is Riparian hermaphrodita. And I called this Tracking Down Virginia Mallow. It's kind of a new title, because I'm mostly going to talk about the fieldwork that I did last summer. So let me just introduce you to the plant. So Riparian is from the cotton family. So that's Malvaceae, for those of you who know it. It's a rhizomatous perennial. And it's actually a globally vulnerable species. So it's extirpated or imperiled in most of the range. That range is the eastern half of North America for the most part. And the rarity of this plant in the mid-Atlantic is one reason that I'm studying it, but you'll soon hear about a couple other reasons as well. But despite the rarity of this plant, it's also a biofuel crop in Europe, which is not something you often hear about rare mid-Atlantic plants, but for good reason. So it can actually grow up to about 4 meters in one season, which is really a significant amount of biomass to produce quickly. And obviously that's a necessity to be a successful biofuel crop. And then this is actually a genus that's particularly interesting from an evolutionary and biogeographic perspective. And I'm just going to review some of that now. So these are sort of the other reasons to study this plant beyond thinking about its rarity and need for conservation measures. So to put it in an evolutionary context, we actually need to think about Malvaceae in the larger sense, particularly, which contains some members you'll know, cotton, cacao. And we need to look at a particular tribe within the Malvaceae called the Malvaceae. So this is a tribe that contains a fair number of really weedy species that you probably know. So cheeseweed is an example, or Malva neglecta. Velvet leaf is another example, or Butylan theophrasti. And also common garden plants, which I'm sure you know, such as hollyhocks and various species of Abutilon and Malva. So within the tribe, there's a really large clade of plants that lack an epicalyx. So the epicalyx is this series of bracts that occurs outside of the first row of the flowers where the sepals are. So it's under the sepals. So unlike most of the Malvaceae, they're actually lacking this. And that's where our particular clade of interest lies. So this clade's informally called the Plagioclase Alliance. And its first diverging lineage, let me just show you here. Hopefully you can see my green pointer, is Riparian hermaphrodita. So it's the very first lineage that comes off in this group that we're calling the Plagioclase Alliance. And then closely related to it are Cyta hookeriana, and then Cyta tenuicarpa, and then 5 other genera, Laurencia, Hoheria, Plagianthus, Gynatrix, and Astrotrichion. I love those names. They always sound like some sort of sci-fi novel to me. So like Cyta hookeriana and Tenuate carpa, Virginia mallow was also once a Cyta. It was Cyta hermaphrodita. But none of these three species actually fall within the proper clade of Cyta. So it creates a taxonomic problem. So in working through the taxonomy for the Southeastern United States, Weekly, who's at the University of North Carolina, and his colleagues began the taxonomic tidying by erecting the new genus Riparian to account for this North American hermaphrodita species. But the two other species remain to be dealt with. So a second major point of interest in the Plagioclase Alliance is in its distribution. So there's this incredible biogeographic disjunction that occurs in the group. So from North America, which is our Riparian hermaphrodita, our subject of interest today, the whole way through to Australia and New Zealand, and then Tenuate carpa is this one outlier, which is an African species. So we've got this disjunction between North America and then basically Australasia with a little African lineage thrown in. So it begs the question of how did this evolve and when? And looking into the biogeography and timing of when and how this evolution took place is work that's still left to be done, but something that's very interesting. So the other major point of interest for this clade is that a number of these taxa are special in that they are part of the roughly 7%, only 7% of plants that are dioecious, meaning that their sexes are separated on entirely different plants. So you have male plants and you have female plants. So then on our cartoon phylogeny here, I've mapped sex expression in these taxa. So you can see here, I've got white as dioecious, black as hermaphroditic, and then black and white mixed is mixed. So it can be some mixture between, somewhere between hermaphroditism and dioecy. So you can see that we've got dioecy in Plagianthus, in Gynatrix, and Astrotrichion. And then you can see some of the flowers here. So I've got Gynatrix as an example here. So you can see the larger male flower here on the right in Gynatrix at the top, and then Plagianthus at the bottom, and then the smaller female flower on the left in both of those species. So then the closest relative to these three dioecious genera is actually Hoheria, which is a hermaphrodite. And then in the biggest genus in the group, which is Laurencia, we see a mixture of these conditions. So we've got some hermaphroditic species, we've got some dioecious species, and then we have some that are even more complicated sex expression. So as far as we know, these three lineages at the bottom are all hermaphroditic, but none of them have been studied in detail. So given the phylogeny, given this tree of life for this clade, it would appear that that rare condition of separate sexes has evolved not once, but multiple times in the Plagioclase Alliance. So better sampling and faster evolving markers for phylogenetic studies are needed to get at exactly how many times it's evolved, particularly in Laurencia, which is a complex group. And then more in-depth developmental and molecular studies are required to tease apart if those separate instances of the evolution of dioecy, they could have thrown different molecular switches each time to enact separate species, enact separate sexes developmentally, or they could have reused a common pathway to repeatedly do the same thing to create the same, the same means to the same end over and over, or maybe they did a mix of these two alternatives. So Jennifer Tate of Massey University in New Zealand and I are currently studying the morphology and the anatomy and the floral transcriptomics of the genus Plagianthus to begin to unravel how separate sexes evolved in this group. So that placement of Riparian hermaphrodita right here as that first diverging lineage in the clade makes it really interesting subject for which to study floral developmental patterns and look for the potential of self-compatibility or incompatibility. So this is whether or not the plant can pollinate itself and produce successful fruit. So looking at floral development in Virginia mallow itself could give us a really good context for understanding floral development in the entire group, such as in the Plagianthus work. And then studying the potential for self-compatibility in Riparian may actually help us start to understand the evolutionary pressures, such as inbreeding, that may have selected for a separation of the sexes in some of those descendants of the most recent common ancestor of this group. So these are some of the reasons that Riparian is interesting beyond just being rare in the mid-Atlantic. So I hope that that's also a good reminder that our mid-Atlantic plants are often globally connected, and to fully understand their context, we really need to stray beyond our regional boundaries. And so with that greater evolutionary context, let's return to the United States and our consideration of Ripariacida and its habitat and its rarity. So for the first part of our study, we conducted a niche analysis to better understand Ripariacida and its distribution and see if we could narrow down the variables that define where it grows. So the first thing we did was to gather data from all the known herbarium specimens of Ripariacida. So this was helped in large part by digitization of herbarium specimens. So we found 247 different specimens that were useful to us, and we georeferenced all of the specimens, meaning that we put a latitude and longitude point on those specimens based on what the locality statement said. So that gave us, then we ranked them into levels of geographic certainty. So we had 51 species that couldn't actually be georeferenced. So there was inadequate information in the locality statement or a complete lack of a locality statement. That meant we couldn't georeference the specimens. And then 83 were georeferenced from general locality statements. So we called those level 1. 106 were georeferenced from precise locality statements. We called that level 2. And then 7 specimens had GPS coordinates that were actually taken at the time of collection in the place where the plant was collected. So this is sort of our gold standard for what we would hope for in terms of geographic data on plant collecting. Unfortunately, we only had 7, but fortunately, they were sort of spread across the distribution of the entire species. So that was, even though it's a small number, at least it was spread out. So we called those level 3. So the result of that work is a map of the distribution of Ripariacida, as I'm showing you here. You can see it's in the eastern half of North America. So the black dots show the roughly 200 specimens that were actually given coordinates and mapped, and then the numbers correspond to 50 putative populations of these specimens that we created by grouping collections within 10 miles of one another. So it's just a hypothesis at what the populations might be. So just one population occurs outside the United States, number 50 up here in Canada, and then the majority of the populations occur here in Ohio and West Virginia, along the Ohio and Kanawha rivers. And then there's one population I actually eliminated from the map that occurs in the fens in Boston, Massachusetts, because it's really highly likely to have escaped from cultivation. All right, so then from those coordinates, we extracted the following environmental data in ArcGIS. So climate, soil, topographic data, land use and cover, and then also proximity to the nearest water and proximity to the nearest transportation. And then we analyze these results defined by those levels of geographic certainty or uncertainty that I talked about earlier, levels 1 to 3. And that was really to better understand the effects of georeferencing uncertainty on the results. So it's not something you see in a lot of studies using herbarium data, and it's a feature that I actually think is sorely needed. So as a researcher who's actually done a good amount of georeferencing, I know how problematic it can be to put points on specimens. And I actually fear that some researchers aren't taking that into enough consideration before making conclusions from their analyses. They sort of download the data and say, well, it came out of the database, so it must be right, without actually questioning how that long point got put on the specimen. Okay, so results. So our first clear finding was that Ripariacida prefers high sun exposure. So in our 200 specimens, we didn't have a single occurrence of a northern exposure. So you can see this here in this graph and whisker plot that has aspect listed on the left here by degrees. And then on the right, I just put the corresponding compass direction. And then we've got on the far left GPS specimens here. So that's our Titus, box and whisker. And then in the middle, we've got a little bit of uncertainty. This is levels. This is level 3 mixed in with level 2. And then on the far right, we've got the most uncertainty added. And you can kind of see that the dispersal of it across aspect is increasing. So there is a potential difference here. There's a much tighter box around the southern exposure with our most geographically accurate specimens, and then less and less tight with the addition of geographic uncertainty. So that could be the result of georeferencing error. But regardless, even with that potential georeferencing error in there are no northern exposures documented. So that we really do see a clear preference for higher sun exposure for Ripariacida. So next, in an analysis of geomorphic categories for these specimens, and here we didn't actually see any differences between the levels of geographic uncertainty. So all the data are presented together. We see that Riparian favors disturbance. So that's both in natural and mechanical disturbance. And we see that because it occurs in floodplains and till plains and road cuts and urban land. And it's also tolerant of both drought and periodic inundation. And we can see that because it occurs in floodplains and stream terraces, outwash deltas, and drainage ways. And then lastly, in an analysis of soil types for specimens, we saw that by and large, Riparian really seems to prefer loam of various different types, and then this sort of ill-defined urban land that exists in these data sets. Okay, so those are the main results from our niche analysis. So what does all that add up to? So let's do a little bit of plant math, right? So if we add the following characteristics, what do we get? Rhizomatous plus fast biomass production, fast growth, plus likes sun and disturbance, plus it tolerates water variability and urban land. So what does that equal? Now, I know if we were all in a room together, that at least some of you Hort volunteers would be shouting out the answer here. But hopefully you're all thinking it equals a weed, right? These are all weedy characteristics. So then really we're left with a single question. How can a species be rare and weedy, right? That seems like two completely contradicting descriptors for the same plant, right? So are there other characteristics of Riparian that might actually be causing it to be rare, despite some other characteristics leaning towards weediness, right? So the first question there, one characteristic that might be at play is fecundity issues. So is there some reason that Riparian is not good at reproducing? So it seems like at least within a population level, it would be unlikely because we know it has vigorous rhizomatous growth. But if we step back and look more at seed production, it could be a bit more of an issue. This would come into play whenever you're thinking about colonization and establishing new populations at least. So then after early reports of low germination in the literature, germination studies in the biofuel sector found that scarification and increased temperatures produced really high germination rates. So it's not completely clear yet if there are reproductive issues that actually occur in the natural habitat outside of, sort of agricultural propagation methods. Alternatively, or maybe perhaps in combination, for an extrinsic factor for why Riparian could be rare, it could be due to man-made pressures on its habitat. So for example, through the widespread destruction of floodplains and riverine terraces in industrial zones in the river valleys where the species occurs in Ohio. So both of these photos were taken from field sites where Riparian specimens were previously collected, but where the species actually no longer exists. But clearly such pressures can only be part of the story because the availability of disturbed land is increasing annually in the United States. And yet, no species can survive total pavement, right? Increasing impervious surfaces. So clearly land use has a really complex interplay with the rarity of this species. All right, so our current work on this species includes conducting fieldwork throughout its range to find new populations where possible, to record precise geographic coordinates with GPS for populations we do encounter, estimate population sizes, and collect tissue for molecular work. And I had the opportunity to begin this fieldwork this past summer. So this is, again, that map of the global distribution of Riparian in the eastern half of North America. And so for the 2021 field season, I really just concentrated on the eastern half of this, so this area here. So let me zoom in on the map so you can see that a little bit better. So we're focusing on Pennsylvania, Maryland, DC, Virginia, and then this one spot in West Virginia. And I revisited all the known locations from which those herbarium specimens that I've been discussing came. So historic localities, and then I had a few newer localities that were found through iNaturalist and other online observation reporting tools. So I was able to visit nearly all the sites, all but two, and this is what I found. So the green diamonds signify where there are present-day localities of Riparian hermaphrodita. So there are currently 14 populations left of the species in the eastern portion of its distribution. So the majority of those localities are found in southern Pennsylvania, and they're all within the greater Susquehanna watershed. And most of these are concentrated up here in the Raystown Lake area, including the two largest populations that I've seen to date. There's been a notable decrease in the populations along the Susquehanna, And then even more so along the Potomac River, which went from over a dozen localities within what is today considered the Chesapeake and Ohio Canal Park to none that are presently there. So from about 14 to 0 in that park along the Potomac. So there's currently a single plant with a single stem, sadly, of Virginia mallow that's on the Potomac River, and it's the only occurrence for this species in Virginia. Likewise, with current data, there's a single locality left in Maryland. Thankfully, it's not a single stem. It's in the west part of the state, and it's wedged between a state highway that you can see right here in this picture and a railroad track next to a closed paper mill. So according to the herbarium specimens, it actually used to occur, the population used to occur on the other side of the road. So at some point, the population jumped the road. It probably actually did so in a culvert that I saw that runs under the road and comes out daylights in the current population. So luckily the population did spread in some time past, because when I was there, you can see here, there's a giant construction project underway where they scraped all of the soil off of that side of the road. So whether there were Ripariacida over there before I got to the site, I don't actually know. So oddly, there is now a fairly robust population, it's number 11 right here, in DC on the Anacostia River. So this is a tributary into the Potomac. So although there were historic populations in DC, they were all on the Potomac side of the city, and most are presently now under where the National Mall is today. And there have never historically been any populations located on the Anacostia. So it's a really interesting population. And the main chore with that conservation will be getting ahead of the invasive plant pressure at the site. So I want to be clear that with a species that occurs in such dynamic habitat, right, these weedy conditions that I talked to you about, high sun exposure, disturbance, maybe with some water dispersal, you would expect some flux in localities, right? You would think, okay, certain populations are going to be blinking out, others are going to be appearing. And this idea of some geographical flux, that's normal, was borne out in a study that was done by Mulholland and some colleagues on a single population, that single one in Ontario that I told you about that exists in Canada, as shown here. So this is a picture of that population in Ontario. And on the right side of the screen here, you can see looping through are colors representing populations of Ripariacida at that site. So she was going out and doing surveys every 2 years as part of her study for exact localities of the individuals in the population. So even if you account for errors in GPS accuracy, it's clear that these subpopulations at the site are moving slightly over time. And new subpopulations are springing up and some other ones are disappearing. And realize that all of that's happening just over 6 years, which is really a very short span of time, okay? So let's look back at our distribution. So remember, the past is in black, the present is in green, with Mulholland's work in mind. So granted, she's looking at a single population level and we're looking at half of the known distribution of the species. But regardless, it's no surprise that we see a change in current day distribution compared to our historic localities, right? Realize that these black dots date back from the whole way back in 1832 was our first collection that we know of Ripariacida, so nearly 200 years ago, through to just a few years ago. But even with that understanding of natural flux for the species being expected, I would say that the 2021 fieldwork that I did on Ripariacida really still indicates a net loss of localities and present day population levels, at least in the eastern half of Virginia Mallow's distribution. So this coming summer, I hope to visit as many of the known locations in the western half of the distribution as I can. So that'll be ranging from Tennessee up to that Ontario population, and then obviously with a lot of time spent here in Southern Ohio and then West Virginia along the Ohio and Kanawha rivers. Okay, so we already discussed the possibility of fecundity being an issue. So we've begun reinvestigating this fecundity question. So meaning, is there something about the reproduction of Virginia mallow that's actually causing it to be rare? So we're studying its germination requirements. So here you can see Kira's hand, she's counting out seeds. And here we're wetting the seeds in a float test prior to scarification and boiling water. And then this is just a close-up of the seeds. They are your typical mallow looking seeds. So then all of those seeds were recently sitting in 3 different treatments. So moist chill, dry chill, or dry room temperature conditions. And the idea was to see which of those treatments produce the best germination rates this coming spring. So you're the first people to see this data. It's hot off the presses. This is pictures I took on Tuesday in the greenhouse. So you can see that this one, this little guy here, is got nice pericotyledons, and its very first true leaf. It's looking very malvaceous already. And hopefully you can see at a glance of the flats of these planted seeds that our germination rates are actually pretty good. So we need to wait another week or 2 for final counts on germination between those 3 treatments. But I can say now that it does not look like there is an absolute stratification requirement or chilling requirement for this species. So it's interesting that we've got these positive results, but it doesn't necessarily mean that there are no fecundity issues for Virginia mallow and its natural habitat. So I kind of hinted at this earlier, you know, just because you can propagate something in a greenhouse doesn't mean that those are the conditions it's seeing in nature, right? So realize too that all 3 of our treatments actually underwent scarification, so meaning that physical breaking of the seed coat to allow germination to proceed for the work. So we did that by this boiling water technique, which is common with mallow. So next year we'll have to look into whether or not scarification is required and some other treatments that maybe mimic the natural habitat conditions more closely. So that lack of scarification in the wild could also still be a possible limitation for fecundity for the species. Okay, so what else? So we're also going to be studying how Virginia mallow grows underground. And we want to answer a couple of different questions with that, such as what does an above ground stem versus a cluster signify? Is there a pattern to how they develop? So meaning, is it always a single stem in the first year? How quickly does it produce these underground rhizomes and new stems? And we're going to be doing this through plantings in a raised bed that we built over on the farm side. And we're going to dig the plants up incrementally to document and measure their growth. Then with the tissue collection side of the work, so this is the other sort of piece of the work, we're going to do population genetic studies of riparian. So this will help us actually achieve a number of goals. And the first is related to what I just mentioned about that underground growth, right? So molecular data should be able to help us distinguish actual individuals. So what may appear to be separate individuals above ground or ramets may actually all be connected underground as a single genetic individual or genet. So this data could help us distinguish how many individuals are in this photo, for instance, or in any given field site or in any given population. So that's just at that sort of individual level. And also the population genetics work could help us illuminate past demographic changes. So thinking about, you know, have there been serious bottlenecks in the history of the species, or has it had a fairly stable demographic history? This could be an evidence if we see fairly low genetic diversity across the population, sort of lower than we would be expecting given the size and accounting for life history characteristics. And then another part of that is any sort of correlation between an inferred demographic change in the past and geography may actually help us illuminate ancestral regions and those newly colonized by the species. Sort of this outstanding question about the species is it native to the eastern half of its distribution, or is it native to the western half of its distribution and it colonized one or the other area, you know, or vice versa? And that's an open question, and we might be able to start to answer that with population genetics data. We have to wait and see. And then a last piece that maybe the population genetic data could help us with is differentiating garden escapees from natural populations. So this would be the idea of confirming or denying if a few outliers in the distribution are natural or not, like this one I discussed earlier in Boston in the fens. The hypothesis here would be escape from cultivation. We know that Arnold Arboretum was growing this pretty early on in the collection history of this plant in herbaria, so that would be a potential route for escape. So if there's one single genotype or just a handful of genotypes for cultivated material, we can make more robust conclusions about escaped versus natural populations in the species. All right, so I'll end here with a photograph of what we currently think is Pennsylvania's longest standing population of Virginia mallow. It was first collected in 1922, so it's about 100 years old this year. You know, and just the fact of this population is really a testament to the complexity of this species, right? I hope I convinced you that it's reasonable to expect some flux in the localities of Virginia mallow and most of its distribution, with populations coming and going. And yet right? And yet we have this population in Pennsylvania that seems to prove that that dynamic nature isn't necessarily a hard and fast rule. So stay tuned for what's next on the Riparian site project, because it's very much a work in progress. And I want to end with a big fat thank you to everybody who's worked on the project. So Dominique Groffman, who was our intern in 2019 and 2020, she did all of the niche analysis work. Kira Matten, our propagator here at Morris, did all of the heavy lifting on the propagation work. I want to thank all those people who also assisted in sharing field locality data, who actually went out in the field to look for plants with me or gave me permission to do so. So at the state level, that would include Rachel Goad, John Kunzman, Steve Grunn, Chris Fry, Johnny Townsend, and John Zuck. And then at the federal level, that's Lane Strickler, Clara Teal, Vita Svanstrom, Andrew Landsman, Maria Sturziker, and Brent Sturry. And then in DC, Damian Ossie and Matt Sarver. And of course, we couldn't have done the niche analysis part of the project without all of those herbaria, which are up here in tiny font, and then all of the federal data sets from which we actually got the environmental and built landscape data. So thanks to all of them. And thank you all for listening, and I'd be happy to take any questions. Just stop my screen share.
Cindy, could you give us a sense, like when you're looking for these, and like in particular that one that was like only one stem of one species?
Yeah.
How can you just describe like when you're in the field and you're looking for this, like the lengths you go to do this, and like what was that in particular, that specific situation like? Yeah,
so the amount that you look for something depends on a lot of things, right? So sort of theoretical things, like you really know it should be there versus you're not really sure and you're just testing the waters kind of. And then also there's also the logistical component, right? So I was trying to cover a lot of ground in a summer. And so for this work, I was doing less of the sort of finding new populations or looking at suitable habitat to try to find new stuff than we would be doing on other projects, like for example, Tim's cryptogramma project, where he really is trying to find new populations, and that's the work. And he sort of, looks for suitable habitat and then goes there to look for the plant. This was very different. I was working from historic localities and trying to see if the plant was still there. In future, I hope to try to get around those areas and see if it moved from there. And I just didn't, it's not in the same place, but it's close by. I did some of that this summer, but I didn't get to do a ton. But like the single stem in Virginia, I'll be completely honest, I walked by it 3 times because it was completely covered in porcelain berry, like head to toe covered in porcelain berry. But I knew it was there because I was in close contact with the federal staff at it's the park across from Chesapeake and Ohio Canal. It's George Washington Memorial Parkway Park, which is the west side of the Potomac near DC. So I actually ended up going out with Maria Sturziker, and she was like, it's supposed to be right here. I know it's supposed to be right here. There's Three Sisters, the islands in the middle of the Potomac. So then we started pulling off, and it was like, oh my God, it's down to a single stem. And this was after a restoration project that they did a couple of years ago. So that one it was just a sad story. Like, I don't know how it got so unmonitored, but they're very understaffed and they have an insane invasive species pressure in that park. Well, along the entire Potomac, to be honest. So yeah, that was a little bit of a particularly sad story, that one. It's also sad because the common name is Virginia mallow. It's never been a major part of its distribution. Like, so it's an interesting one, but yeah. I hope that answers your question, Brian.
Absolutely, that's good.
Cindy, did you see any plants, were all the plants hermaphroditic that were flowering that you saw?
Yeah, so that's a good question. So there is something going on with Riparian and I haven't figured it out yet. And you can ask Tim how much I've been thinking about this because he has to hear it ad nauseum. But there's something strange and I'm not entirely sure what it is. But I have some plans to figure out what it is. But what I've seen flowers where they have exerted stigma. So we're in Malvaceae, right? So you've got all of your anther filaments, your stamen filaments are fused into a tube. And then you've got your styles coming up from within that tube. to actually, see the light of day and be exerted or be available for pollen capture, right? So I've seen flowers with the styles exerted and open anthers, so typical hermaphrodite, what you'd expect. Then I've seen flowers with the styles exerted, anthers chock-a-block with pollen, but not open. And this is like late in the season. So that's interesting. And then I've seen anthers with completely open, dehisced, pollen everywhere, and the styles are nowhere in sight. So I would say, okay, we've got something complicated going on here. We've got males, we've got females, we've got hermaphrodites. Like that's what I would go with on the floral thing, but it's not quite right because I have yet to see an epicalyx. So I have yet to see a calyx that doesn't have a fruit in it. So I'm not sure what's going on. And yeah, like I said, I've got some plans to sort of figure out how to look at that. And I'm very excited about it because if there's something strange about it, then it puts that whole context of sex expression evolution in that Plagioclase Alliance that I showed you at the beginning into a much more interesting picture, right? So yeah, great question, and stay tuned because I don't know yet.
Yeah, Cindy?
Yeah, hi Hiram.
In Europe, where it's used as biofuel, I guess they must grow it in huge fields and somehow get it to grow, harvest it, move it to storage, and eventually burn it. Is the total environmental effect of doing all that beneficial compared to coal? I know this wasn't part of your study, but is it still a good thing to do, or are they trying to get away from it or increase it? Do you have any feeling for that?
No, I don't have a great feeling for that. And I'll even say the biofuel literature on this is slightly hard to decipher. So this is mostly being done in Eastern Europe. So most of the papers are coming out of Poland. They're not always clear on exactly. There is actually a lot more in what you're getting at, which is sort of like the fuel efficiency and a lot of the sort of analysis of like carbohydrate loads and like the actual chemical composition of what's in the plants as a biomass and how it burns and how efficiently it burns and that sort of thing. I'll be honest, I haven't read those papers because they're kind of beyond what I'm trying to get at here. But yeah, in terms of like its pollution levels and that kind of stuff, I don't have a great idea about that. No, sorry.
I have one more question. I don't want to monopolize, but is there any evidence of this in the fossil record?
Not that I know of. Yeah. But it's, I feel like that kind of hits on this question of what has it always been rare? And to all indications, just from herbarium records, that's true. But that's the last 200 years. That's not fossil record. But my inclination would be to say it's always been rare. And that if that's the case, then you're much less likely to see it in the fossil record. But we'll see.
But if it's connected with species in Australia, New Zealand, it's had to have been around a long time, I would think, right?
Yeah, it can't have been. Like we know the date for Malvaceae, we know the date for things that occur, diverged well before this alliance, and it's got to be long distance dispersal between those three areas. That much I can tell you for sure from just the context of what we know about timing for the evolution in the cotton family. Other questions? Has anybody ever seen this plant? Sorry, Jen.
No, it's okay.
No. Hands up, nobody's ever seen it. You might see it because we have literally like 1,000 of them in the greenhouse. So Kara was like, so what are we going to do with all these? When I talked to her on Tuesday, I was like, that's a really good question. So she was suggesting, yeah, we'll see. We'll have to decide what to do with them. But go ahead, Jen.
Do you guys actually do the genetic testing here, or do you do any genetic extraction or anything? Or
so some people might think I paid you to ask that question, but we're going to be doing it very soon when we get our lab. on the
Awesome.
farm side. We're waiting for, we're hopefully getting a modular trailer and we'll start doing it. I have the skills to do it, but I don't have the lab to do it. So yeah, we're very excited about starting to have the capability to do that. And I have a whole bag of samples in the freezer waiting.
When you start doing the genetic testing, are you planning to go to Europe and take samples of their
It definitely
biofuel?
needs, yeah, definitely need some biofuel.
You need any help, you let me know.
Have you always wanted to go to Poland, Pam?
Sure.
Yeah, right.
Oh, Chef's,
I've heard Krakow's nice. Yeah.
Go to New Zealand if you want to go somewhere.
Yeah, yeah. So I mean, so the background on this is I did my postdoc on Plagioclase in New Zealand. So that's part of why that's a big part of the story for this for me, because the sex expression part it's really interesting and it's not common. So I think that's going to be this whole companion piece to the sort of rarity conservation part of this study. So yeah, especially if it's not a hermaphrodite, right? So then, you know, I already have a name for that paper. Worst plant name ever. Riparian hermaphrodita is not a hermaphrodite, right? Like it's easy. Nor does it really occur in Virginia anymore. It's like the worst plant name ever. Anything else? Well, thanks, guys. That was really fun. Even if it wasn't in person.
Thank you so much, Cindy. It was really fascinating.
Yeah, it was.
I'm not sure if I'm the MC for the day, but thank you everyone, or Pam if you are. But yeah, thank you everyone from everyone here. Yeah, that's great. And I think we're going to leave, if I'm not mistaken, folks asked to leave the video up so people can chat. So if for volunteers to chat with each other, so we can do that for you guys to hang out for a little bit.
Yeah, that's fine. I can always, I'll end it. I think Liza may have made me a co-host, so I can end it if people wanted to stick around and chat a little bit. That's fine. If people want to do that.
Yeah, Brian? Can you hear me?
Yeah. Yep.
Can you give us an update on the spring course catalog?
Yeah, actually it should be hitting your doorsteps any moment. The classes go live Friday morning at 9 a.m. So you'll be getting that. I mean, I think Jerry got it at her house last night, so it should be hitting soon. It'll be live, yeah, Friday morning.
Okay, thanks.
And we have a new, and we can never replace Jan. But Jan's replacement will be here March 1st. So yeah, so we're going to get, yeah, new classes, new things. So looking forward to that as well.
I have a question for Cindy.
Yeah.
When you're looking for rare species such as one of your focus, does one always look in the areas that have historically had these? Does one ever look somewhere else, far afield, use the pun for that?
Yeah, so you start with the historic places, like that's definitely the starting point. But then, you know, once you have a feel for where you're seeing it, what the habitat type is, then you branch out, right? If you have the time and the money, hopefully. Right. And I will say, I actually found a couple new localities. So one of the ones I found was about a mile upstream of a historic locality. And I just went there because, you know, I was frustrated because I hadn't seen anything that day. And I was like, oh, I'm just going to try over here. And I like literally parked my truck and got out, and there was one right next to my truck door, which was really convenient, but and surprising. So it was a single plant, but still, you know, it's interesting because nobody's seen it there since like 1950 or so. So yeah, so you do branch out from those historic localities after you kind of get your feel for it. Like I mentioned the cryptogramma work that Tim has been working on for a couple of years. He's really got it narrowed down to where he's actually pinpointing localities on GIS based on particular environmental conditions, right? So in that case, there's much more specific requirements as a plant. It's not such, it doesn't sort of have this weedy thing that Riparian does. It's a little bit more fussy. So yeah. So the answer to your question is yes.
And speaking of GPS, so when you mentioned that there are 7 of them that have been GPS located, so were those the most recent ones, and that's why? Because as we get further into the 21st century, we're getting more with it. Was that why?
Yeah, they weren't exactly the most recent ones, but they were sort of, we don't really start to see a lot of GPS data on herbarium specimens until you get sort of past 2000. Some people were doing a little bit earlier than that, but mostly, it really ramped up like, after the 2000s. So yeah, they were more recent collections. And then of course the iNaturalist data and the actually had, it was a Maryland rare plant observation reporting tool. I had some data from that. Those are just GPS points. You don't have specimens to go with them. Sometimes you have an image. But people were super helpful. The woman who found this location in Maryland through this sort of citizen community science app, she got on the phone with me. She was like talking me through it because, there was a strangeness about creek names and, the locality that she'd given me that makes sense. And she ended up just calling me. I mean, it's just super helpful. People are really interested in kind of reaching out.
These days, I would imagine she could just send you a picture over your phone and say, to show you, this is what I saw.
Yeah, right. But I was actually trying to go back to the site and, I think, it was great. It was very nice to see how interested people were.
As an amateur, but a state employee in New Jersey, I was involved with, because part of my job was to look at wetlands and wetland delineation. That was involved with Helonius ballata, swamp pink, which is a federally rare endangered species, and it has some populations in southern New Jersey. And part of my thing was to look for those, and occasionally I would see them and, just tell the applicant, sorry, or, 300-foot buffer or something like that.
Right.
Stay away, but.
Yeah.
And during my tenure there, one of my colleagues did find a new little population somewhere. Here I was around Camden County someplace. But back in the 80s or 90s, I think, something like that. So that was fun.
Yeah.
Yeah.
Definitely exciting. I think it's going to be strange to sort of shift gears from being in the eastern half of the distribution where, you sort of, unless you're up at Raystown Lake, it's sort of few and far between and a little bit depressing. And you're like, dragging your legs through Japanese hops and just wondering where these plants are. To then go to the western half this coming summer, I think when I'm in Ohio, West Virginia, it's like, some of the locality statements on the herbarium specimens, like all along Route 26, up and down the Ohio River as far as I can see. So it's like, I think it's going to be night and day, and the question is, why is it so locally abundant in one corridor, right? But nowhere else. So I don't know the answer to that yet, but it's something that keeps me awake at night.
And would that all be floodplain or?
Say it again.
Would that all be floodplain there you're dealing with, or?
Yeah, I mean, it's all riverside for the most part. We do have some instances. This was a strange one. We had one instance of it being on this mountaintop in Pennsylvania that I found out about really late in the summer. And I have no idea why it's there. The only connection to sort of its typical habitat is that they just did a road project up there and they had brought in a lot of equipment. So I'm wondering if seed came in on that equipment, because I don't have any other explanation, because it's nowhere near water. It's nowhere near a source population. Like, it's just really up on this mountaintop in state game lands, not near anything. But luckily they noticed it was something different, the guy who manages that game lands, and he flagged it and reported it to the state. And so it's interesting, it'd be interesting. It'd be really interesting to see what the population genetics, if we have any data sufficient to sort of tell us what that might be most closely related to.
Are they wind pollinated or do they need insects and big yellow equipment?
Yeah, it would be insects, not wind.
Not wind.
But I'm not totally sure on what the insects are. I saw bees visiting for sure, but I don't know. I think they might be fairly generalist. I don't think, they're not orchids. So, but yeah, definitely some sort of insect mediated pollination.
Cindy, when do you typically try to go out there and look for them? Like, because you're probably trying to see them when they're in flower.
Yeah, I
would
did last
guess.
summer. I focused on the flowering time because I hadn't seen it much. But I think this summer I'm going to branch out because they start to come up like April. And then, they're pretty much done by like, now it's probably Thanksgiving, but you could say end of October. But I think I'm going to, just because of the amount of sites I'm trying to get to this summer, I think I'm going to have to spread it out and go before flowering time, which is July. They flower for at least July, and they flower for a long time, like almost the whole month.
Okay.
They flower sort of in a sequence, and then you even start to see fruit production sort of mid-July, and then the fruits are on through like end of October. So it's, there's a nice wide range. This is no day flowering thing. So that's pretty easy, but it's not so critical now to see especially now that we have so much material in the greenhouse, it's not so critical for me to see the flowering stuff in the wild. So I really just need leaf samples. So we're even thinking about trying to hit the Tennessee populations over spring break so that I can just knock that one off, because it's just such a large amount of ground to cover, but we'll see.
Right. Do you plan to also collect seed then when you come across it in the fall so that you can do other?
Yeah. I need to decide about that because there's definitely more propagation questions that we have, but I'm not exactly sure what the best way to source them is. Like, I did some things this fall to collect seed to sort of get at that question that Bill hinted at, this, okay, is it truly hermaphrodite? So I was really specifically collecting leaf material from the parent and then single fruits from the fruits of that parent so that I can get at sibling relationships genetically from the maternal parent, because that will help me start answering questions of selfing versus not.
Right.
Hopefully. But I don't know. Like, in an ideal world, I would have spent this winter getting those genetic data and I would know the answer and then I would proceed with the next field season, but I can't do that. So I'm kind of guessing, but it's not that hard to get to Raystown from here. And that's an easy source. I have good connections with the people there. The populations are large enough. They're not going to be hurt by me sampling fruits. So I think that's probably going to be the main source for propagation work for a while. We could also just grow up the 1,000 plants we have, and then I'd have all the seed I need forever. So I'm not sure.
Also, my other question had to do with associated species. Like, when you found it, what were some of the other associated species at that time? Or was that pretty variable based on the invasives? So all the Potomac sites are pure invasive almost, except for the trees, right? The canopy is still native. There's a little bit like that single stem of Virginia had a nice Vernonia noveboracensis growing right next to it. So it's not strictly invasive, but that's what's growing over it, pulling it down. And it's the really obvious part of what's growing with it. A lot of Japanese knotweed competing a lot, because it's almost the exact same habitat, except for Japanese knotweed can also grow in the shade, right? So it's like really similar riverside, high disturbance. Like, so that was probably the number one. And then, I'm just trying to think of the more natural habitats. So a lot of them were mown field, sort of like intermittently mown field. Like the ones up around Raystown, the Army Corps of Engineer sites, and so it's your typical field stuff, right? Like Solidago, it's not right.
It's not some really rare habitat. It's not some uncommon mix of very specialized plants at all. It's
But most
high
of it's
sun
pretty
exposure,
disturbed,
yeah.
right? So
Yeah,
it's
and the
disturbed full sun, and
disturbances-
so that's the invasive, you know,
Yes, the disturbance
niche.
thing seems to be the only commonality amongst all of those sites. So, and it's interesting that one I mentioned on the Anacostia, that one in DC that came up. So they found it in 2012, and it's a dredge island. The whole island is dredge, and it's a recreational site. But they had bulldozed, you know, maybe in the 70s or something, to build a soccer field that never got finished. So it's a high disturbance area too, even though it's kind of a recreation site. And they think that that might have been related to when it started to come up, and then it was sort of discovered, you know, just about 10, 15 years ago or something.
Right.
But
So it has
it's a big population, like it's flourishing. So it'll also be really interesting to see what that's related to. But, you know, like the historic record for DC area, it was all on the mud flats that are now the National Mall. Like, you know, the herbarium statements, the locality statements are like 18th and Northwest. Like it's like literally like halfway through the mall, where it used to be tidal flats. Like this is probably back in the day when it was like tidal flats and sewer outlets, you know, like.
Right,
so,
right.
Yeah. So, I mean, it's interesting, but we'll see.
So it's
Yes.
possible that the seed bank has a really long life, which is why once disturbed,
I don't
you're
know.
not sure.
A seed bank question, Tim and I were just talking about that. I don't have a great sense of seed bank longevity. But maybe we can figure that out. I don't know. I need to just stick some. I would actually, there's two sites that are, so that Raystown stuff, you know, so you've got where they put the dam in and made Raystown Lake. All the sites are north of that. So they're downstream of it. But south of there, it used to be a large area. Like there was like 7 or 8 sites. And now there's a single plant. And that's that new one I found that was a mile away from the historic record. And there's 2 sites there that are on power line right-of-ways, which is a really common place for these plants. Waterside, Riverside right-of-ways. And I'm just slightly convinced that, it's a weird pairing of words, but that if I just dug up like 4, you know, cubic feet of that soil and brought it back here, that we could grow some riparian. Like I'm not convinced it's gone from there, right? Like if we could just like get the Japanese knotweed out or like bulldoze the Japanese knotweed, I just wonder if you would see some riparian come up, you know? But who knows? I don't have a great sense of the seed bank potential. So we need like 10 years to start to get at that question, or 100 years. So I don't know what the answer is there.
Cindy, at one of the Larry Wiener's conference, a ecological landscaper out in York County redid that golf course that kept flooding. Do you remember that? And they ended up taking about 30 feet of silt off of the back to get it back to normal creek levels. And then they found old seeds germinating.
Yeah.
From that. So, and that was what, a couple 100 years the seeds survived in the, anyway, so that's been talked about other times.
Yeah.
So it can, but I wanted to bring up INAT. You know, I know you had sponsored us going out and using iNaturalist during the citywide challenge. And,
yeah, the City Nature Challenge. Yep.
Thank you. Yeah, and so I just learned that our master gardeners across the state of Pennsylvania are going to have the option to learn and use iNaturalist to record pollinators on all types of pollinators, not just butterflies, on certain pollinator gardens to feed into a research study. And so I just wanted to mention there's a population of users that might, you know, that's going to expand, which is kind of good to hear. So it's, you know, Penn State research, entomology.
Right.
Okay, just,
cool.
Thank you.
But I don't mean to monopolize the chat room. So if you guys want to chat amongst yourselves, I can. I'm sure it's nice to see each other when you don't get much time to do that right now. But anyway, thank you, everybody. And it was nice to see your faces, even if not in person.
Thank you, Cindy.
Thanks, Cindy.
So Cindy, you and Tim are still co-hosts, but Pam is not. But it looks like Liza's staying on. So now Tim Block and Liza are the only co-hosts.
That's fine. Liza's technically still on. She might be in another meeting though. Hey, Bob.
Hey, Pam,
Yes, Hiram.
who's collecting the timesheets for last year? Can we give them to you or? you know?
Yeah, you can send them to me. Can you scan it in and email it, or do you want to just mail it in? Whatever is
I'll drop it off
easiest.
next time I'm there,
Okay, that's fine. That
unless you
sounds
need it
good.
quicker.
No, it's okay.
All right, thanks.
Yep.
All right.
I like seeing your cat, Andrew.
Yeah, he has something about my iPad. He's always, might have to do with the fact that Greg and I will listen to a Great Courses birding series on it. And
Oh, he's hoping
occasionally
to
there'll be
catch.
bird calls and he goes crazy over that. So
That's funny. Bye, Bob.
I'm gonna say bye too.
Bye,
Yeah.
Andrew.
Yeah.
Bye, Hiram.
Okie doke. Well, I'm gonna call it then.
Sounds good.
Thanks for playing our game. Good to see you all.
Thanks, Liza.
Uh-huh.